morning, dear student. Today, in this section, we will learn about the composition and structure of DNA. What is this DNA? DNA is a nucleic acid. So, how many different types of nucleic acids exist? Two types. They are DNA and RNA. DNA means deoxyribonucleic acid and RNA means ribonucleic acid. Then where we can locate these nucleic acids? In the case of eukaryotes, DNA is confined. It is located inside the nucleus. We can't see any DNA outside the nucleus in the case of eukaryotes. But in the case of eukaryotes, taking the case of RNA, it is commonly, that word we need to notice, commonly seen in the cytoplasm. That means that in some situations, some type of RNA we can locate inside the nucleus also. That is understood there. So commonly RNA is seen in cytoplasm in the case of eukaryotes. In the case of eukaryotes only. Then, what about prokaryotes? Prokaryotes, we know it doesn't possess any nucleus, so the genetic material is found freely in that cell itself, and it is called a nucleoid. So that's why regarding the prokaryotes is not mentioned here. So anyway, we can move to the next point, that is about the occurrence. Both the DNA and RNA togetherly present in most of the organisms. But in some organisms, some exceptions are there where either RNA or DNA only is present. One example I am mentioning here is the tobacco mosaic virus. In this virus, the genetic material is RNA. So it is an RNA virus. Another example is T2 bacteriophage. Its genetic material is DNA. There is no RNA in the case of this bacteriophage. That's about its occurrence and existence. And then we need to understand what are the building blocks of this DNA? What forms the DNA or what forms the nucleic acid? It is formed of monomeric units of nucleotides. So many nucleotides, just like beads in a string, it forms the nucleic acids. So, nucleotides is the basic building block of this nucleic acids. And again, a single nucleotide, even it is composed of so many things. Which are those things? They are nitrogenous base. We can write it like that. Nitrogenous base is there, a pentose sugar and a phosphate group. Okay, BSP. And the sugar that present in the nucleotide is a pentose 5-carbon sugar. So these are the these are the components that form one nucleotide. That one after another, they together form the nucleic acids. Okay, then among this, if we look to this nitrogenous base, it is also different types. Which are the different types of nitrogenous bases we can see in DNA and RNA? They are adenine, guanine, cytosine, and thymine. In the case of DNA, and among these different nitrogenous bases, why I wrote this thymine only in red color? And that is one important thing. So that we will understand as we look to the names of the nitrogenous bases present in RNA. In the RNA, all these bases exist, adenine, guanine, cytosine. But instead of thymine, another base present, that is uracil. So there's a difference between DNA and RNA. What difference? Other difference also there. What difference is that among the nitrogenous base in RNA, instead of thymine, uracil is present. Then all these nitrogenous bases again we can classify it into purines and pyrimidines. Purines and pyrimidines. All these purines are double ring structure, two ringed structure. I will show you. Purines, they are two ring, double ring structures, and purines are adenine and guanine. Okay. For easy, we can remember it as poo. By that way, we can memorize it and not get confused with the type of pyramids. So, purines are adenine and guanine, and they are double ringed structures. But pyrimidines, they are single ring structures. And which are the pyrimidines? Cytosine, thymine, and uracil. And need to notice that this uracil, we know that it present only in RNA, and thymine, it can be located. Oh, sorry, it can be seen only in DNA. Okay, so that's it. Then in what way we can indicate all these bases? By using its first letter. Adenine by A, guanine by G, cytosine by C, thymine by T, and uracil by U. Okay. Now we clearly understood what is nitrogenous base, which are the bases, how it is classified, everything. So next element in the nucleotide is sugar, pentose sugar. So that's what we are going to see now. Pentose, the word itself indicates that sugar is a 5-carbon sugar, and that of two types. Which are those two types? Deoxyribose sugar and ribose sugar. Deoxyribose sugar is the sugar that present in the DNA, and ribose sugar is the sugar that present in the RNA. What is the difference between these two types of sugar? This picture is shown here. Look to this picture. In deoxyribose sugar, deoxy means oxygen is removed. Oxygen is removed. So 2 is the position. So this is the second carbon. In deoxyribose sugar, oxygen in the second carbon is lost, it is removed. So that is why deoxyribose. So the sugar that present in DNA is deoxyribose sugar. While at the same time that sugar, ribose sugar that present in RNA, it possesses that oxygen in the second position. So it is simply ribose sugar that is present in RNA. So pentose sugar also clear to you now. Okay. So we finish nitrogenous bases and pentose sugar. Next element is present in nucleotide is phosphoric acid. Phosphoric acid is the same one in both RNA and DNA. There is no much difference in the case of these two nucleic acid. And the importance is that this phosphoric acid give acidic nature to the nucleic acid. Otherwise, the nucleic acid is basic. So, acidic nature is provided by the phosphoric group that present in the nucleotide. Next, we are going to learn about what is nucleosides and what is nucleotide. Nucleotide diet, I already explained to you that it is the monomeric unit that forms the nucleic acid. Then what is this nucleoside? This nucleoside is a combination of base, nitrogenous base plus sugar. What is lacking here? Phosphate group is lacking here. So, without the phosphate group, that forms the nucleoside. And which is the bond that is holding the base plus sugar? It is the glycosidic bond. I will show you that glycosidic bond. This is the sugar and this is the base. This is the sugar and this is the base. This is pyrimidine and this is purine. Okay. In the case of the pyrimidine, if the base is pyrimidine, the nitrogen in the first position of the pyrimidine, it forms glycosidic bond. This is the bond, glycosidic bond with the sugar, first carbon of the sugar. And if that base is a purine, It's ninth nitrogen is forming glycosidic bond with the first carbon of the sugar. So that's it. Nucleoside. The nucleosides are of two types. Deoxynucleosides that is present in DNA, and ribonucleosides are that one present in RNA. For example, deoxyadenine, that is the nucleoside present in DNA. Deoxyadenosine. Deoxy. I will write here. Deox. Deoxyadenosine. In the case of ribonucleoside, what would be this one? Simply adenosine. So that is the difference. Then coming to nucleotide. Nucleotide means it's very simple. We already have seen that what are these elements along with this sugar one phosphate group also coming, then it become nucleotide, nitrogenous base plus pentose sugar plus phosphate group. Okay, and by the addition of this phosphate group, that convert the basic nucleoside to an acidic one. And which is the bond that is holding that nucleoside to that phosphate group? It is the phosphoester bond. So phosphoester bond is the bond that is connecting that phosphate group with the nucleoside. Nucleoside, we know it is base plus sugar. So I will show you that. So here either a purine or pyrimidine. Commonly we can call it as a base. So base will be there, and it is attached with the sugar, pentose sugar, and this pentose sugar is phosphoester. Through phosphoester bond, it is connected to a phosphate group. So this is the structure of nucleotide. So here base is there, then sugar is there, and phosphate group is there. So that all together form the nucleotide. Which are the types of nucleotides? They are deoxynucleotides and ribonucleotides. We know that deoxynucleotides are the one that present in DNA, and ribonucleotides are the one that present in RNA. And example for deoxynucleotide, we know we can take the case of this one itself. We know that along with this as one phosphate also join, it become nucleotide. So deoxynucleotide, its example is deoxyadenosine monophosphate. So in what way we can write? Deoxyadenosine, this much form the nucleoside. Then monophosphate. In the case of RNA, it becomes adenosine monophosphate. So these are the different types of nucleotides. Okay, I think it's clear to you. Next, we are going to understand about polynucleotide. Poly means so many nucleotides. How they are connected? They are connected in the form of a chain. So many nucleotides connected in the form of a chain with the help of phosphodiester bond that will form a polynucleotide. So it is a chain formed by linking nucleotides through phosphodiester bond. Then what is this phosphodiester bond? Phosphodiester bond we can see in this picture. I will show you that. Here, one phosphoric acid one phosphoric acid is connected to two sugars using that phosphodiester bond. This bond and this bond. Two ester bond. So that will form the phosphodiester. Phosphodiester phosphodiester bond. This phosphoric acid is linked with the two ester bond, phosphodiester bond. By that way, it links two sugars together. 5 dash group of one sugar is linked with the 3 dash group using a phosphoric acid. Ester bond, ester we know it forms between alcohol and acid. That OH group, alcohol group is provided by this sugar, and acid group is provided by this phosphoric acid. So that's through that way, ester bond is formed here. Two ester bond that connects two sugars of the polynucleotide chain. So that is phosphodiester bond. Okay. So polynucleotide is that chain formed by linking of nucleotides through phosphodiester bond. And what is phosphodiester bond? That also now very clear to you, I hope. Then what about the position of the nitrogenous base? Nitrogenous base in this picture we can see here. First carbon of the sugar, deoxyribose sugar, it is connected to the nitrogenous base. And also all these bases, they are oriented at right angle to the long axis of polynucleotide chain. I will show you that. So this is the polynucleotide chain and its long axis we can see. And if we consider that axis, the direction, orientation of that nitrogenous base, it is right angle to that long axis of the polynucleotide chain. So that orientation is right angle. Okay. So that nitrogenous base, it is positioned right angle to the long axis of polynucleotide chain, like steps in the ladder. Like steps in the ladder. Next point is about the polarity of the chain. For understanding that, I will show you an image. This is a portion of DNA, and in this picture we can see two polynucleotide chain, and the orientation of one chain is one chain ends up in 5 dash phosphate group, while the other one ends up in a 3 dash hydroxyl group. That means that the orientation of these two polynucleotide chains are opposite. So polarity is opposite polarity. So that's regarding the polarity of the two polynucleotide chain. And then we need to understand about nucleoside triphosphate. For example, ATP is a nucleoside triphosphate. Adenosine triphosphate. What is this? What is its role in forming the nucleic acid? Actually, this nucleoside triphosphate, all nucleoside triphosphates are the precursors of nucleic acid synthesis. How it acts as a precursor? That I will show you in an image. So this is a portion of a polynucleotide chain, this much area. And here we can see one nucleotide triphosphate. Here one sugar is there, base is there, and three phosphate group is there. And the end, 3 dash end of this polynucleotide chain, 3 dash end of this polynucleotide chain, it interacts with this phosphate group and form an ester bond connecting these two sugars that we can see here. So that phosphate group is incorporated and it is used up to form the phosphodiester bond to connect these two sugars and released these two phosphate groups that we can see here. And that released two phosphate groups are called pyrophosphate. So in that way, nucleotide triphosphate, they are the precursors of nucleic acid synthesis, nucleoside triphosphate. That's about the polarity and the nucleoside triphosphate. With this, we can finish today's class and section. In the next section, we can learn about Watson and Crick model of DNA. Thank you.
I'm an icon, man. I'm a girl about a 10. I should fuck her in a Benz.